So we are still continuing under the what? Under the cell division, because the meiosis, which we talked about what? Double A and double B, right? We have talked about what? The combination of both, which is what? AB, that gives us back to what? 46 chromosomes, right? And I have said that what? This is what? Diploid. They are diploid in nature. What means by diploid in nature makes that what? They are always in foreign numbers. They are not haploid. Because another A will come here, 46. Another AB will come here, 46. Another AB will come here, 46. They are diploid in number. 1, 2, 3, 4. That's the reason why we call it diploid in number. Do you get that? Yes, but that is not exactly where we are going. That's not exactly where we are going. Where we are going is that in meiosis, the process by which the production of reproductive cells, gametes, are having the half number of the chromosomes of the parent cells. And that's what in meiosis, there are two divisions. One, first meiotic division. Second meiotic division. There are two processes like that. Remember, I've talked in the previous videos that what? That for a diploid is 4, right? But this division has to take in two places: the first division and the second division. What is first division? First division occurs in the what? In the parent. Right? And second division occurs in that what? In that gamete itself. Do you understand what I mean by gamete? What they actually produce. I mean by that the gamete. This is the parent cells, right? This is the parent cell. The 46 chromosomes of the male and the 46 chromosomes of the female. They both what? Give half 23 pair to form another 46 chromosomes of a gamete, right? You know that with what? What do you call it? A cells have been from 46 is now 23 here. From 46 is now 23 here, making 46 once again. Another what? Another chromosomes, 46 chromosomes is being formed, right? Now this 46 chromosomes, this is what? This is the combination of different cells from the egg and from the sperm and the egg, which is now give us the gamete, right? The second division is now taking in that gamete itself. The second division is now taking place in that what? In that gamete. And that's the reason why, aside from the division, they have this series of processes. Series of processes. Yes, they have series of processes. In meiosis, they have series of processes. When the division is actually occurring, along the way, the processes is going from one to one. One from Prophase1, Prophase 2, Metaphase 3, Anaphase 4, Telophase. But be reminded that these are what? 1, 1, 1, 1. Prophase 1, Metaphase 1, Anaphase 1, Telophase 1. This is the first series. Now the second series is what? Prophase 2, The same thing, metaphase 2, anaphase 2, and telophase 2. There's their series. This is the first one that when the division begins to start. This is the first series that will come into play. The Prophase 1 will come into play, followed by the Metaphase 1, followed by the Anaphase 1, and the Telophase 1. Once this series is completed, it's now coming back to Prophase 2 rather, Metaphase 2, Anaphase 2, and Telophase 2. What is now the difference between each of these series of stages? The series of Prophase 1 to Metaphase 1, Anaphase 1, Telophase 1, Prophase 2, Anaphase 2, Metaphase 2, and what? Telophase 2. What is now the difference between them? Let's discuss about the word. Each functions in the process of what? Meiotic division or meiosis. Meiotic division or meiosis. The same thing, if I say meiotic division, I'm talking about the word, the production of the abscess of the gamete from the chromosomes of the parent. When I say the meiosis, it's just the process. So, okay, let's now talk about the word. About it. Okay, so we talked about what? About the cell division. But the Telophase 1, the Prophase 1, what's their own function in the cell division of meiosis? Right? Let me give an example of this before I proceed to the work. This is, let's assume this is one chromosomes. This is the second chromosomes. Right? Okay. And these are another chromosomes from the. I decided to use two different colors so for us to understand. Right? Good. Let us now call this the word. A, just we have been the same before, and this is B. Right? A for father or for male, and B for the mother. Right? Okay. Now let's come back to the Prophase. A process is a process whereby what? The chromosomes A and B align. They align beside each other. Align beside each other. Right? How? A and B. A and B. And another A and another B. You see, they are now different to this one. That's the reason why I want to discuss. Even if it's at all, we can discuss a little or three phases here. The first series, then we'll be discussing the second series in the other subsequent videos, right? One of the two has come to align with one of the two here. That is when they are aligned beside each other. This comes into play, and also this comes into play. The second one comes into play, and the second one this process is called synapsis. Let me write synapsis. Right? The process by which one of the chromosomes pair with the another chromosomes, and the second chromosomes pair with the second chromosomes of B is called what? Synapses. Now we are having BA and AB here, and BA rather, BA, because we have named our green as our B and our black as our A, same here, right? But it's not, their work has not yet finished. What is next is that in these chromosomes there is something called chromatid. I'm using another color which is black, right? Which is blue, right? The reason why I'm using this color is for us to understand chromatid. Chromatid. This is their chromatid. Yes. The synapses occur exactly at the point of the chromatid. They align at the point of the chromatid because there will be a what? Cross pairing between each of them to produce the what? The new generation of what? Of another chromosome. You know, this is chromatid. One chromatid brings us to a full chromosomes. Another one chromatid to another one chromatid brings us to a full chromosomes, right? So there is a point here which is called the chromatid. The chromatid. So at this point there is what? A crossing. There is a crossing. Now let's see that crossing. No, sorry please. And this is the point. Can we get that? Yes. Same thing for the other one. You know, this is for A, and same thing for the other one. Good. Good. You are good to go. Yes. This is what? This is the full chromosomes. We are now having the full chromosomes. How? Because they have cross breeding here. Cross here. Same here, crossing occur. Same here. Good. But if you are to go a bit far from here, in chemistry they usually say there's an hydrogen bond here, but you don't need that. There's an hydrogen bond here that makes them to join together. When they are talking about the bonding in chemistry, they talk about about the hydrogen bond that usually occur in gene, and this is exactly where it occurs. But we are not going there. So this is exactly what occurs in Prophase 1. This is only what occurs in Prophase 1. The first one is that they align together in the process of what? In the process of what's called synapses. In that process, they don't align ordinarily. They align at the point of their chromatid. Each of the chromatid has chiasmata. Sorry, please. Chiasmata. This is exactly what I want to say. Chiasmata. Their point. Each of these is called chromatid. But the point by which both of them join this point and this point is called chiasmata. Similar to this chiasmata. That's the reason why I'm using different pen to write it, different paper to write it. Chiasmata. So at the point of their chiasmata, there is crossing. There is crossing. Similar to this. To produce what? New chromosomes. You know, this is now 46 chromosomes in full. This is now 46 chromosomes in full. If you were able to check it, this is now the first thing that occurs. You know, we talked about that it is of two divisions. It is of two divisions, right? We said the first meiotic division occur in parent. The second meiotic division in our previous video we talked about the division of the meiosis. We said the division of meiosis is occur in two stages. The first meiotic division, which occur in parent, and the second meiotic division, which occur in that garment itself. In that garment itself. And this is exactly what happened in the what? In Prophase 1. Now, let's now proceed to Prophase. So rather, Metaphase 1. Metaphase 1. What occurred in Metaphase 1? We have already said that it is a series. That means that what? When the first stages finish, the second stages carries from where it is finished. So it means that where the Prophase finish in the crossing, there's the point of what starting from the metaphase. The starting point for the metaphase is the end point of the word Prophase. So let's now do what? Let's now discuss the metaphase. So let's now discuss what happened in the what? In the metaphase. Like in the metaphase 1. So what occurred in Metaphase 1? We have already said that the end of the word, the end of the stages of Prophase 1 is just the beginning of the stage of what? Metaphase 1. Good. Okay. We have talked about that. You know, you have the diagram like this, right? Okay. We are good to go. Right? Okay. Good. So what is now going to occur here is that each of these chromatid has their own spindle. What do I mean by spindle? Spindle. Spindle. They have their own spindle, right? This spindle is like a, let me say, it's like a length of a wire that is going to attract the north pole and the south pole. Okay. Now, these want to react. These want to react with this, right? So they begin to form. I call it spindle. I don't call it a spindle. They begin to form this. So there's a reaction like this. There's a reaction like this, right? So similarly, this we want to react with this. So there's a reaction like this. The spindle wire is trying to connect each other, right? This is exactly what happened in the metaphase. The spindle in each of the chromatids is trying to react with another spindle in the other chromatid. These are already formed a chromosomes, but they still want to, because everything is occurring in something like this. Just let me do it like this. Everything is occurring, so it's like a magnet. The more they see, okay, there's another opportunity for me. They have formed a chromosome. They see, okay, there's an opportunity for me to react with this again. So they begin to form this what? This spindle, and this spindle occur in this metaphase. Now, once the spindle occur in the metaphase, that is this is the function of the metaphase, just to let the stage at which the spindle of each chromatid try to react with the other spindle, and that being goes to what? To the anaphase. That being goes to the anaphase, because in this, there's a reaction between them, then something like this again. Sorry, this thing is not writing up. Okay, good. Let me use this. Then each of them trying to come to the equator. This is black. This is okay. This is another one. Good. You can see they start from Prophase where they combine, right? Now into the Metaphase, the spindle formed. The spindle formed here, and now both of them come to the equator of the what? Of the nucleus. They stand here. The other one also stand there. They are broken again here, just like they are in the first place. It's not that they are broken in the Prophase. There is a reaction, but the reaction doesn't end there. The reaction continues in the Metaphase. But in the Metaphase, they see, okay, we can break again. They break, and each of them stand at the equator, trying to prove that what there is a process which is meiosis process, which bring the haploid of each to bring the diploid, of the chromosomes. Okay, let me try re-explain from Prophase. Prophase is a point at which the chromatid, each chromatid align side by side. That point of align is called the chiasmata. And there's what? Synapses, the process by which they align to each other. The process, just like distorting, the process by which they align together is called what? It's called the synapses. But the point at which they align, each of them align together, is called the chiasmata. After the alignment, at the point of the chiasmata, there is a crossover. The crossover brings us to this place. That's the stages of Prophase 1. Now, from Prophase 1, we come to Metaphase 1, right? The Metaphase 1 now thinks that there is a further reaction that can occur in each of the chromatid. So they bring the spin. Each of them have different spindle. So they bring their spindle out from a reaction to occur. That reaction occur, and they find out that okay, we can still break. That brings us to the Anaphase. Now they break. The green here has joined this is the green here. The green here has come to this extent. The black here has come here. The black here has come here, and the green here has come here. That brings us to the face of Anaphase. Do we get that? Yes. After it brings us to the face of Anaphase, now what actually occurs in the Anaphase is that they have already occurred. Okay. It's not giving us that this and this occur here because they have occurred here. Let me say the telophase. Okay, the telophase. Please don't mind. Let me try and clean the board. So, the telophase, just like the diagram we have been bringing for ourselves. Good. So, we know we have talked about that what in Anaphase they have come to the equator, right? Good. They have come to the equator. So this is exactly what occurs in the Anaphase. Before we proceed to the Telophase. Okay. What now occurs is that in the Telophase, you know they have their own nucleus. The nucleus of this now comes. A very big nucleus of this now come into play. Because they cannot be continue forming this process occurred every time as the division is going into the cell. That's why we said what the process by which the production of the half cell of equal half cell or half number of the chromosomes of the parent is called what is called the meiosis. This process begin every time. So this is their nucleus. Both the nucleus of what of this and the nucleus of this, which is this and this. Now contrast. It constricts, right? What do I mean by constrict? That there's a formula like this, something that is bigger now try to constrict, just to make that way. Okay, let's complete this reaction. Let us complete this reaction. And at this point, both of the let me say the upper side and the lower side bring down their nucleus, and their nucleus now constricts just to make the continuation in the mitosis. You wonder I said mitosis? Yes, because this continuation doesn't end here. After it stopped here, they go into the interphase. But we are not going there right now. But that one is in mitosis. The next thing we're supposed to go is what is the. Let me try and explain this again once again. Each of these chromosomes, they have their own nucleus. This one also have their own nucleus. At the point of what, okay, we have been trying to do the reaction several times. Okay, the nucleus of this now comes to nucleus of this. They have to constrict just to produce what? The daughter cells. We usually call it daughter cells. Just to produce the daughter cells, which is what? You know, this is what? Haploid. This is haploid. So the combination of these three haploids now give us what? The full diploid. The full diploid. Because the daughter cells, which is what? 4 in nature. If you count 1, 2, 3, 4, which is being produced, there is a reaction here that is being formed from the nucleus of which, when they constrict, they have to produce what? The diploid cell. They are forming the haploid cell only before, but you could remember that I said what? In the daughter cell, the gamete that produce in the meiosis, they are diploid in nature. They are not haploid, but the process whereby the haploid process has to occur before the diploid process has to be completed. And that's exactly what happened in the what? In the Telophase 1. Yes, Telophase 1. So at this point, we'll be discussing later from the what? Prophase 2, Metaphase 2, Anaphase 2, and Telophase 2. So in our subsequent video, we'll be discussing from the what? From where we stop from the Telophase 1 and begin with what? Prophase 1, Metaphase 1, Metaphase 2 rather, Prophase 2 rather, Metaphase 2, Anaphase 2, and Telophase 2. Thank you. Don't forget to subscribe to this particular channel. Thank you once again.